Hello and welcome to Bibliophil's e-classroom. In this lecture, I am going to talk about what is a vaccine and the various types of vaccine and the various stages involved in the development of a vaccine. The topic of vaccine today has been elevated to a significant position because of the prevalent COVID-19 pandemic. As you know, countries all over the world are competing today to invent the vaccines for COVID-19 as soon as possible. And recently, India and the United States, in their joint effort to combat the COVID-19 pandemic, are planning to work together to jointly develop a vaccine for COVID-19 under the Vaccine Action Program, that is VAP. This Indo-US Vaccine Action Program, that is VAP, is a 33-year-old collaboration between the two countries. Under this collaboration, various research institutions of both the countries are involved. It includes the National Institute of Health, that is NIH, from the United States, and from India it includes the Department of Biotechnology and the Indian Council of Medical Research, that is ICMR. Through this Vaccine Action Program, both the countries are jointly working towards developing a vaccine for COVID-19, and they are also planning to jointly test the vaccine candidates as a part of the clinical trials. So in this context, let's understand what is a vaccine. But before knowing about vaccine, we need to get acquainted with some basic key terms which we are going to come across frequently in our discussion. So firstly, we need to know what is a pathogen. See, in simple terms, the microorganisms which have the ability to make us sick is referred to as the pathogens. This includes the usual bacteria and viruses and also some protozoas and fungi. For example, SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus 2, is the pathogen responsible for the disease COVID-19, which is the acronym of Coronavirus Disease 2019. And then strep throat is a sickness caused by the pathogen Streptococcus bacteria. And the pathogen of malaria is the single-celled protozoas. Now, what is antigen? Generally, the human immune system cannot recognize the pathogen as a whole. The immune system actually recognizes only a specific part of a pathogen that is present on its surface. Antigens can be any type of molecule, that is protein, carbohydrate, nucleic acid, or even fat. Meaning of antigen is antibody generators. So once our immune system recognizes these antigens, it induces the immune response system in our body to produce antibodies. And antibody is a specialized Y-shaped protein that can help the immune cells to destroy the antigens. Antibody is also called immunoglobulin. Now, what is immune system? Immune system is the basic defense system of the body that protects us from harmful pathogens and diseases. Humans have two types of immunity. One is innate and second is adaptive or acquired. Now, the innate immunity is like the natural immunity. Everybody is born with innate immunity, which is a type of general protection. For example, the skin of human body acts as a barrier to block the germs from entering the body, which is a type of innate immunity. And then the adaptive immunity, which is also referred as the active acquired immune system, it develops throughout our lives. We develop adaptive immunity or acquired immunity when we are exposed to diseases or when we are immunized against them with vaccines. So having discussed these key terms, let's now understand what is a vaccine. See, vaccine is essentially a biological preparation which provides with immunity against certain types of infectious diseases that can be caused by virus, bacteria, etc. A vaccine provides us this immunity by training our immune system to identify the infection and fight the infection. Since this immunity is not naturally present in human, and since it is acquired through vaccines, this immunity is referred to as active acquired immunity. The vaccine actually trains our immune system to acquire this immunity. See, whenever a foreign pathogen enters our body, generally the immune system recognizes it as a threat, and this activates the immune system to produce antibodies which fight against the foreign pathogens. Generally, this is how our immune system protects us against infectious diseases, but there might be a few bacteria or viruses which may not be recognized by our immune system. It may be that our immune system has never been exposed to them. The best example for this would be the SARS coronavirus 2. Since it is a novel coronavirus or a new coronavirus, it is not being recognized as a threat by our immune system. And hence, the immune system is not getting triggered, no antibodies are being produced, and hence we are not able to fight against this infection. So this is where a vaccine comes in. It trains our immune system to identify the new pathogens as a threat and produce the required antibodies. So essentially, the vaccine is providing us with active acquired immunity. Now, let's talk about the classification of vaccines. See, broadly, there are 4 types of vaccines. These are whole pathogen vaccines, subunit vaccines, toxoid vaccines, and recombinant vaccines. Now, the whole pathogen vaccines can be subdivided into 2 parts, that is live attenuated vaccines and inactivated whole cell vaccines. And similarly, the subunit vaccines can be subdivided into 3 types, that is protein-based vaccines, polysaccharide vaccines, and conjugate vaccines. Now, let's look into these vaccines in a greater detail. At first, I am going to talk about the live attenuated vaccines, which is a type of whole pathogen vaccine. See, live attenuated vaccines contain the whole pathogen, that is the whole cell of bacteria or virus, which have been weakened so that they create a protective immune response but do not actually cause the disease. These wild viruses or bacteria are actually weakened by repeated culturing in cells in which they do not reproduce very well. Attenuation can often be achieved by growing a pathogenic bacteria or virus for prolonged periods under abnormal culture conditions. And as these live attenuated vaccines are so similar to the natural infection that they help prevent, they create a strong and long-lasting immune response. Just 1 or 2 doses of most live vaccines can give you a lifetime of protection against the germ and the disease it causes. But live vaccines have also some limitations. For example, live vaccines are not suitable for people whose immune system does not work well, either due to drug treatment or any underlying illness. This is because the weakened viruses or bacteria can multiply too much and might cause disease in these people. So if these vaccines are administered to a person who has an impaired immune system, for example, they have leukemia or HIV infection, or they are taking immunosuppressing medications, administration of live attenuated vaccines may cause severe disease in them as a result of uncontrolled replication of vaccine virus. Now, the examples of these live vaccines include the tuberculosis vaccine or BCG vaccine, which is a bacterial vaccine. And among the viral live vaccines, there are oral polio vaccines, which is also called the Sabine form of vaccine, as the scientist Albert Bruce Sabine developed this type of vaccine, this oral polio vaccine. Then the other viral live vaccines are rotavirus, yellow fever, measles, mumps, rubella, that is MMR combined vaccine, etc. Now, the second type of whole pathogen vaccine is inactivated whole cell vaccine, that is the killed antigens. This type of vaccines contain whole killed pathogens. They have been killed or inactivated through some physical or chemical processes. Inactivated vaccines are not active and cannot replicate, so there is always a need for multiple doses. Inactivated vaccines usually don't provide immunity that is as strong as the live vaccines. So one needs several doses over time, that is the booster shots, in order to get ongoing immunity against the diseases. In general, the immune response does not occur after the first dose, but usually after the second or third dose. And as inactivated vaccines do not contain any live bacteria or viruses, they cannot cause the disease against which they protect, even in the people with severely weakened immune system. So that is the advantage of these inactivated vaccines. And examples of inactivated vaccines include whole cell pertussis vaccine, that is a bacterial vaccine, and also the inactivated poliovirus vaccine, which is a virus vaccine. And this polio vaccine is also called Salk vaccine because this vaccine was developed by Dr. Jonas Salk, which came into use in the year 1955. So here you can see the polio vaccine are of two types, that is Sabine vaccine and Salk vaccine. And the Sabine vaccine came into the use of in 1961 and the Salk vaccine came into use in 1955. So actually the Sabine vaccine replaced the Salk vaccine, and generally the Sabine vaccine is more effective than the Salk vaccine, and the Sabine vaccine is given orally and the Salk vaccine is usually given by injection. And actually the Sabine form of vaccine has played the key role in nearly eradicating the disease from the world. And there are also some examples of inactivated vaccines are Hepatitis A, rabies vaccine, etc. So now let's talk about the second broad type of vaccine, that is the Subunit vaccine, which is also called the acellular vaccine. Acellular means not containing any whole cells. So subunit vaccines do not contain any whole pathogen, that is whole bacterial cell or virus cell. So subunit vaccines contain polysaccharides, that is sugars or proteins from the surface of the bacteria or the viruses. And these polysaccharides or proteins are the parts that our immune systems recognizes as foreign, and they are referred to as the antigens. See, subunit vaccines, just like the inactivated whole cell vaccines, do not contain any live component of the pathogen, but they differ from the inactivated whole cell vaccines by containing only the antigenic parts of the pathogen. And these parts are necessary to elicit a protective immune response. Like the inactivated vaccines, subunit vaccines are considered as very safe. However, subunit vaccines often require the incorporation of adjuvants to elicit a strong protective immune response because the antigens alone are not sufficient to induce adequate long-term immunity. See, an adjuvant is a substance that is co-injected with antigen in order to help stimulate and enhance the adaptive immune system into producing antibodies against the antigen. Adjuvants is added to a vaccine to boost the immune response to produce more antibodies and longer-lasting immunity. And it also minimizes the dose of antigens needed. Now, the subunit vaccines can be further categorized into protein-based subunit vaccines, polysaccharide subunit vaccines, and conjugate subunit vaccines. So firstly, the protein-based subunit vaccines are made using a specific isolated protein of the pathogen, and this protein component provides or acts as an antigen and induces the immune system of our body to produce antibodies. Examples of protein-based subunit vaccines are acellular pertussis and hepatitis B vaccines. See, the first whole cell inactivated pertussis vaccines introduced in the 1940s comprised inactivated B pertussis bacteria. Although very effective, whole cell pertussis vaccines frequently caused minor adverse reactions such as fever and swelling at the injection site, and this caused many people to avoid the vaccines. And by the 1970s, decreasing vaccination rates had brought about an increase in new infections. So then, further research led to the development of acellular pertussis vaccines that are based on individual purified B pertussis components. These vaccines are similarly effective as wholesale vaccines but much less likely to cause adverse reactions. And then the hepatitis B vaccines are composed of the hepatitis B virus, the hepatitis B virus surface antigen, a protein which is produced by the hepatitis B virus. Now, the second type of subunit vaccines are polysaccharide vaccines. See, some bacteria when infecting humans are often protected by a polysaccharide capsule, that is the sugar capsule, that it helps the organisms to evade the human response system, especially in infants and young children. And polysaccharide vaccines are a unique type of inactivated subunit vaccines composed of long chains of sugar molecules that make up the surface capsule of the certain bacteria. Polysaccharide vaccines create a response against the molecules of the pathogens capsule. Presently, pure polysaccharide vaccines are available for 3 diseases, that is pneumococcal disease, meningococcal disease, and Salmonella typhi. Now come to the third type of subunit vaccines, which is conjugate subunit vaccines, which is the most important subunit vaccines. See, conjugate means connected or joined. It was found that the polysaccharide vaccines did not work well in the infants and the young children. So researchers discovered that they worked much better if the polysaccharide was attached or conjugated to something else that could create a stronger immune response. So in most conjugate vaccines, the polysaccharide is attached to carrier protein. A carrier protein is actually a protein linked to a weak antigen to increase its immunogenicity when used as a vaccine. See, various protein carriers are used for conjugation, including diphtheria and tetanus toxoid. See, toxoid is nothing but the inactivated toxins obtained from a pathogen. I have talked about toxoid later in greater detail. So conjugate subunit vaccines can prevent common bacterial infections for which plain polysaccharide vaccines are either ineffective or provide only short-term protection. Actually, the advent of conjugate subunit vaccines heralded a new age for immunization against the diseases caused by encapsulated microorganisms such as meningococcus, Haemophilus, influenza type B, and pneumococcus. See, the HIV vaccine or the Haemophilus influenza type B vaccine contains a polysaccharide joined to tetanus toxoid. The first licensed vaccine against Haemophilus influenza type B was a polysaccharide vaccine. However, its usefulness was limited as it did not elicit strong immune response in infants. And then the researchers developed a conjugate vaccine in which the HIV polysaccharide is attached or conjugated to a protein antigen to offer improved protection. This formulation greatly increased the ability of the immune systems of young children to recognize the polysaccharide and develop immunity. And then MenC vaccine or meningococcal C vaccine gives us protection against meningococcal disease, and this vaccine contains a polysaccharide joined to a tetanus toxoid. And then comes the pneumococcal conjugate vaccine, that is the PCV vaccine, which contains polysaccharides from the surface of 13 types of bacteria which causes pneumococcal disease. It is joined to the diphtheria toxoid. And the PCV7, PCV10 and PCV13 are important pneumococcal conjugate vaccines. And then comes another type of conjugate vaccines, that is MenACWY, that is meningococcal group ACW 135 and Y conjugate vaccine. This vaccine gives protection against 4 types of meningococcal disease caused by group A, C, W and Y. This vaccine contains polysaccharides from the surface of these 4 types of bacteria which causes meningococcal disease joined to diphtheria or tetanus toxoid. So now comes the third broad type vaccine, that is the toxoid vaccines. See, some bacteria release toxins, which is actually poisonous proteins, when they attack the body. The immune system recognizes these toxins in the same way that it recognizes polysaccharide or proteins on the surface of the bacteria. Some vaccines are made with inactivated versions of these toxins. They are called toxoids because they look like toxins but are not poisonous. Toxoid is actually the inactivated or killed toxin used in the vaccine production. Usually, the toxoid vaccines trigger a strong but short-term immune response. And to increase the immune response, the toxoid is absorbed to aluminum or calcium salts, which serve as adjuvants. And examples of toxoid vaccines are diphtheria vaccine, tetanus vaccine, etc. Now let's talk about the fourth important type of vaccines, that is recombinant vaccines. See, the recombinant vaccines are made using bacterial or yeast cells to manufacture the vaccine. A small piece of DNA is taken from the pathogen, that is the virus or the bacteria, and this is inserted into other cells to make them produce large quantities of active ingredients for the vaccine. For example, to make the hepatitis B vaccine, part of the DNA from the hepatitis B virus is inserted into the DNA of yeast cells. Then these yeast cells are then able to produce one of the surface proteins from the hepatitis B virus, and this is then purified and used as the active ingredients in the vaccine. Now, examples of recombinant vaccines other than the hepatitis B vaccine include the HPV vaccine, that is the human papillomavirus vaccine, and the MenB vaccine, that is the meningococcal B vaccine. Today, scientists are developing and evaluating recombinant vaccines to protect from viruses such as HIV, Zika virus, Ebola virus, etc. So these are the various types of vaccines which are currently being administered worldwide. See, we can attribute many public health success to vaccination. For example, the worldwide vaccination campaign eradicated smallpox, polio in most of the countries. And also, the childhood vaccination has substantially reduced the morbidity and mortality from infectious diseases. However, the future presents continued challenges. Still, there are many diseases for which researchers have been unable to find effective vaccines, such as HIV, AIDS, malaria, etc. Also, in some cases, the cost of vaccine is too high for the poorer countries. And although many of the current vaccines are highly effective, efforts continue to develop vaccines that are more effective than those available today. Thus, researchers continue to explore new possibilities. Scientists are still working to create new types of vaccines. So in this regard, let us look into the future of vaccination. See, the development of nucleic acid-based vaccines, which is also known as the DNA vaccines, is currently being explored. DNA vaccines consist of DNA coding for a particular antigen, which is directly injected into the muscle. And then the body's own cell use this genetic material to produce the antigens. This type of vaccine has the benefit of being relatively easy to produce, since DNA is very stable and easy to manufacture. But this is still in the experimental stage because no DNA-based vaccines have been shown to elicit the substantial immune response required to prevent the infection. Although many such DNA vaccines are in the research pipeline, none are currently licensed for human use. But researchers are hopeful that DNA vaccines may be able to generate immunity against parasitic diseases such as malaria. Currently, there is no human vaccine in use against a parasite. Researchers has also developed candidate DNA vaccine to address several viral diseases threats, including SARS coronavirus 1 of 2003, H5N1 avian influenza of 2005, H1N1 pandemic influenza of 2009, and Zika virus of 2016. Then, the vaccines based on messenger RNA, that is the mRNA, are also being developed. See, mRNA is an intermediary between DNA and protein. Recent technological advances have largely overcome issues with the instability of mRNA and the difficulty of delivering it into the cells. and some mRNA vaccines have demonstrated encouraging early results. For example, researchers developed an experimental mRNA vaccine that protected mice and monkeys against Zika virus infection after a single dose. Most importantly, to counter the prevalent COVID-19 pandemic, the US-based biotech company Moderna Inc. is developing an RNA-based vaccine, which is being called the mRNA-1273. And the company has already successfully completed the phase 1 trials of its vaccine candidate. And it has also been given the go-ahead by the FDA to begin the phase 2 trial, and moreover the company is supposed to begin the phase 3 trials in the month of July, whose results may be available by the end of this year. These are all you need to know regarding vaccines and various types of vaccines. So in this session, we have learned various key terms like pathogens, antigens, antibodies, immune systems of human body, and what is vaccines and various types of vaccines. And we have also learned the future of vaccines. And in the next part of this lecture, I am going to talk about the various stages involved in the development of a vaccine and other important things pertaining to vaccines. So this concludes our discussion. Thanks for watching.